For today's webinar, we have Joni Squaya, Product Application Specialist with InnoGenomics, Dr. Erin Hansen, Research Associate Professor at the National Center for Forensic Science at the University of Central Florida, and Justina Nichols, Research Scientist at the Center for Forensic Science Research and Education, presenting Interlaboratory Comparison of SpermX and Conventional Differential Extractions. Additionally, today's presenters will be joined by Dr. Jack Valentine, Associate Director for Research at the National Center for Forensic Science during the Q&A. Without further delay, here's Joni, Dr. Hansen, and Tina.
Hello everyone, I'm Joni Squaya, and we'll be discussing an interlaboratory comparison of SpermX and conventional differential extractions. The development and validation of the SpermX method was recently published in the journal Forensic Science International Genetics, but I will give a brief overview of the method before discussing the interlaboratory study. The key to the SpermX differential extraction is the incorporation of a matrix membrane. It is nanotechnology-derived polymer separator, and you can see from the slide on the right, which is a scanning electron microscopy of the membrane. The membrane will capture the sperm cells and only allow the digested epithelial cell DNA and other non-sperm DNA to pass through the membrane. It's a rapid and reproducible system and can be automated. The SpermX methodology consists of placing the swab into the inner tube. That swab remains in the inner tube throughout this whole process, unlike the conventional differential extraction where the swab is removed after the epithelial digest, which can result in the loss of a lot of sperm DNA, which I'll discuss later in the presentation. The swab stays in for the first epithelial digest to help remove non-sperm DNA. The sperm cells get trapped on the matrix membrane. Hence, there's no technical skill needed from a qualified DNA analyst, and a technician can be trained to do this. You do not have to separate and pull off your supernatant from your sperm pellet as you do in the conventional differential extraction. The two epifractions that are collected in the SpermX method are called E1 and E2. Again, the swab remaining in the tube for 3 washes, and also remaining in the tube for the final sperm digestion and collecting of the sperm fraction DNA, which is usually referred to as the S fraction. The mixture samples used for the study will be shown on the next slide. I encourage you to take a screenshot when I get to the next slide so you can have that for reference for the remainder of the presentation. The mixtures consisted of 8 swab sets of female to male mixtures. The female component was saliva and the male component was semen. The biological fluids were pre-quantified prior to get the concentration of DNA, which was determined to be approximately 21 nanograms per microliter for the saliva donor and approximately 36 nanograms per microliter for the male semen donor. From those concentrations, known amounts were prepared to give ratios as shown in the chart, ranging from 100 female to male down to 18,182 parts female to 1 part male. Each mixture consisted of triplicates for the SpermX method and triplicates for the traditional differential extraction method. The 3 laboratories that participated in the study is InnoGenomics, who processed the swabs with the exception of A through D for the differential extraction method, and the University of Central Florida and the Center for Forensic Science Research and Education processed all 8 swabs in triplicate for both methods. QIAGEN chemistry was used for all purifications. InnoGenomics used the manual QIAMP investigator kit, and UCF and CFSRE used the EZ1 DNA investigator kit. Each of the 3 laboratories used their own in-house conventional differential extraction method. InnoGenomics followed the manufacturer's protocol for the manual QIAMP DNA investigator kit, which has the lysate, buffer, and alcohol ratio as 1:1:1, and elution in 60 microliters. University of Central Florida used the QIAGEN EZ1 DNA Investigator Kit for their conventional differential extraction. They used the procedure per the manufacturer's protocol, which has 190 microliters of buffer G2 and 10 Pro-K for the epi digestion for 1 hour, and 160 of the G2, 10 of the Pro-K, and the addition of the DTT for the sperm digestion incubation, which is 10 minutes at 70 degrees, and then uses the EZ1 trace protocol for an elution of 50 microliters. CFSRE also used the QIAGEN EZ1 DNA Investigator with several modifications from what UCF had used. They replaced the epithelial digest buffer with their own EZ1 extraction buffer. The recipe is shown over to the right, and that's a fairly common epithelial digest buffer. They've lowered their incubation time for the epi from 1 hour to 40 minutes, and they also increased the 10-minute sperm digestion runtime up to 40 minutes, using the same trace protocol with 50 microliter elution volume. For quantification, IGT used our own InnoQuant HY on a Quant Studio 5. UCF and CFSRE used Trio on a 7500 and a Quant Studio 5 respectively. For the amplification and typing, IGT ran Paraplex Fusion Kit on a 3130, analyzed with GMapper IDX version 1.4. UCF and CFSRE used Global Filer on 3500s with version 1.6 of GMapper IDX and version 1.4 of GMapper IDX respectively. Dr. Tim Califat ran the StarMix method using generic StarMix parameters to correspond to the STR kits and stutter values and instrument platform, which I described previously, using each of the lab's individual analytical thresholds. The analytical threshold used by InnoGenomics was 100 RFU, UCF used 100 RFU, and CFSRE used 40 RFU, just as a reminder. And all StarMix runs use the same proposition, being hypothesis 1 being the female and the male semen donor, and hypothesis 2 being a female and an unknown individual. And the LR was determined using the HPD with a lower bound. Also came out with template amounts and donor proportions for all the runs. The metrics for successful sperm cell separation from female epithelial cells were evaluated for the sperm fraction quantifications. They include the male DNA recovered, that is the total male that is found in the sperm fraction for each of the methods, total human to male ratios, which will give an indication of how much female there may still be in the sperm fraction, the male recovery percentage relative to the theoretical amount that was originally deposited on the swabs, and the percent female carryover in the sperm fraction. The data was culled from all laboratories, and metrics for successful sperm cell separation from female epithelial cells were evaluated. The first of which is the male DNA recovered in the sperm fractions. For the first set of mixtures, A through D, you can see the average total Y and the standard deviations from the triplicates. SX is represented in the blue bar, and the differential extraction is represented in the green pattern bars, and that will be the theme throughout the discussion. You can see that UCF and CFSRE exhibited more male DNA captured with the SpermX device and method than their conventional differential extractions. Note, InnoGenomics did not run A through D with DE, so there is no comparison. Mixture swab sets E through H are what I refer to as the low sperm series, because in each of these mixtures there's a constant 1,500 nanograms of female DNA with 2.5 nanograms, 1.25 nanograms, 0.33 nanograms and 82.5 picograms male in each of these mixers. You can see the trend that occurs when you go from E being the 1500 with 2.5, then the 1500 with 1.25, 0.33, and then 82 or so picograms. You could see the decrease with the low amounts of male given the same amount of female. And all three labs showed the blue bars or SpermX method giving higher male DNA yields than did the differential extraction. The SpermX method resulted in higher yields of male DNA for all the samples, with the fold difference ranging from 1.5 to 12.6 times more DNA. You could see the range in the chart for all the mixtures, A through H, with the average fold over all the mixtures being that SX recovered 5 times more male DNA than did the DE. The SY ratios in the sperm fractions were determined using the small autosomal for the trio kit and the short target for the InnoQuant HY kit. The short target is divided by the respective Y targets of each kit, and less than or close to 1 indicates a good separation. In this graph, you see the average of the triplicates and the associated standard deviations. You can see that the average SY ratios were all lower for the SpermX method in blue than they were with the differential extraction in the green. This is indicative of less female carryover with SX compared to the DE. And note that there were no standard deviation bars for the differential extraction for H because all three labs had only one replicate, hence no standard deviation. The fold difference in the SY ratios can be seen in the chart at the right. You can see them individually for each laboratory and then the average of all three laboratories. The DE-SY ratios went from 5.4 to 183 times higher than the SX, again indicating that there's more total human target that the DE has relative to the male, and that would be indicative of a poorer separation of your male and female DNA with the DE method. Another metric used was the male percentage in the sperm fraction, and in this case it's relative to the theoretical. So we had a known amount that was put on the swab, and now we're trying to see what is the percentage recovery of male DNA that resides in the sperm fraction. And in this case, you can look at the graph on the right, and you're going from A down to the low sperm series on the right, E, F, G, and H. And you can see that SpermX recovered significantly more male DNA than did the DE, and the fold more male DNA increased as you go to lower sperm. You can see the very small green bars relative to the larger blue bars on the right-hand side of the graph. And you can see that G and H, the very last two, with the 100 cells and 25 cells, show the highest fold differences between the methods. And looking at the fold difference in the male percentage recovery, if we look at the first set, A through D, we can see that SpermX went from 1.5 to 10 times more DNA. And if you look at E through H, you can see that SpermX recovered from 20 to over 1,500 times more DNA. And that's specific to G and H. You can see it was 124 times and 1,533 times more DNA as you went to very low sperm samples. The percent female carryover that resided in the sperm fractions was calculated by taking the amount of female DNA found in the sperm fraction over the total female DNA. For SpermX, that is 3 fractions, E1, E2, and the sperm fraction. And for the differential extraction, it consisted of an E and an S fraction. So this is not to the theoretical, but what is the percentage female that was in the sperm fraction relative to all the female detected from the swab. And you can see that the DE exhibited higher percentage female carryover than SX for every set for each of the laboratories. The higher female carryover for the DE method ranged from 2.8 to about 10-fold more than the SpermX. The A mixture was removed as CFSRE had an outlier there of 112 times more, leading to an average of 57.9. Overall, the large amount of female carryover then results in more complex mixture interpretations with very low or minor male or loss of the male allelic information. The STR profiles were evaluated to determine the detection of the male component in the sperm fractions. Basically, how much male information was obtained in each of the sperm fractions for both methods. We did that manually by looking at the number of unshared male alleles recovered. And it would be, for the different kits, it was 31 for the fusion kit, a number of unshared male alleles, and there were 32 unshared male alleles in the global phyla kit. So it was literally a count of if you had 30 out of 31, it would be a high percentage. And if you got 5 out of the 31, obviously a lower percentage. And that was all done manually. The data was then analyzed by Dr. Tim Califoot at Sam Houston State University, run through SpermX. And the metrics that came out of that was the male profile proportion. That is how much male-female DNA is in the profile. For example, if it was a 50-50 mixture, or 70% male, 30% female, etc. And then the statistics for inclusion of the sperm donor are provided as a likelihood ratio. I will be presenting the STR metrics for the InnoGenomics lab, and then we'll have our colleagues from the other labs go over their data. The STR profiles were provided from one of the 3 replicates. We normally chose the sample that had the lowest SY ratio to give the better yields for both methods. As I mentioned, there's 31 unshared male alleles in the fusion kit for these 2 particular female-male mixtures, and the recovery of the unshared male allele was evaluated and shown as a percentage. The average at the bottom is just for G and H, since it was 100% for all the other mixtures. You can see that SpermX recovered the unshared male alleles in a full 100% in 6 of the 8 mixtures, and in E and F, both the SX and the DE recovered the full complement of unshared alleles. Again, this is just a count of the alleles, not anything to do with the peak heights or any other type of probabilistic genotyping at this point. Mixture G resulted in 30 male alleles and for the SX method and 17 for the DE method. For mixture H, the SX method gave 18 alleles versus only 1, which was a homozygote for the male component in the DE method. This is a graphic representation of what you just saw in the chart, showing that you got the full complement of unshared alleles for both the E and F series. Now keep in mind that this entire series had 1,500 nanograms of female, and the E fraction had 2.5 nanograms. The F fraction had 1.25 nanograms. So when you had over a nanogram, you were able, with the differential extraction, get the same number of male alleles that were not shared with the female. But when you get down to the G series, where there's only 330 picograms, and the H series, where there's only 82.5 picograms, that's where you see the differences, where the differential extraction is not able to get as much male allelic information as does the SpermX method. The blue channel electropherograms are shown for mixture G, with the SpermX electropherogram on the left and the conventional differential extraction on the right. Bring your attention to the amelogenin locus, the first locus there, with the red square, showing that the SpermX had an X of 6139 RFU and a Y of 473, which is a X to Y of about 13-fold, whereas the DE at amelogenin X was 8471 and the Y was 105. So you can see the X is higher and the Y is lower, and that one has a X to Y of about 81. So you can see there was a lot more female in the differential extraction for G than there was for H. Additionally, you could see the arrows showing where there was male alleles called for the SpermX method, and they were missing from the DE method. I also have them circled so you can see the RFU levels that were not detected by the conventional differential extraction. Now, this is just one channel. Overall, The G gave 30 out of the 31 alleles, whereas the DE gave 17. And the next electrophotogram, I will speak about the H mixture. For the H mixture, this is the one that had the 82 picograms of male DNA to 1,500 nanograms of female DNA. And for the SpermX method on the left, showing the yellow channel, we saw that there was, for the entire profile, there was 18 alleles that were detected in the SpermX method, whereas in the differential extraction method on the right, for the entire profile, the only allele that showed that was foreign to the victim was that 29, which happened to be the male homozygote genotype. Now note, I don't show all the other colors, but there was no Y whatsoever at a melagenin, and there was no DYS391. So in the entire profile, there would just be one indication that there was a donor other than the female herself. You can see on the left-hand side, I've circled some of the alleles that were present in the yellow channel for the SpermX that were not present in the DE method, just to give you an example of one of the channels of the electrophotogram. SpermX results included the male profile proportion. You can see here, SpermX is in blue, and the differential extraction in green again, and you can see that the low sperm samples from E through H, where the comparisons to the DE were made for InnoGenomics, showed a 2.8 to 4-fold more male being observed in the SpermX STR profiles than in the DE profiles. And the differences for each one are shown in the chart on the left. For the SpermX analysis, the statistics were done such that the genotypes of the female and male components were provided, treated like a sexual assault kit, such that the female would be the victim and the male would be the suspect. The mixture samples got treated as if they were evidentiary sexual assault samples, and then had the calculation done as an inclusion statistics. That gives a measure of the amount of information that's gained from each of those profiles to then make the comparisons between the two methods. You can see in these results that, again, we say 10 to the 27th, but remind you that septillion is 10 to the 24th and octillion is 10 to the 27th. We never had to count that high till we got such high likelihood ratios in our random match probabilities in forensics. But the SX resulted in higher LRs for the low sperm series. You can see that the order of magnitude ranged from 2 to 16 orders of magnitude, such that there was significantly more male or more information gained from the male component with the SpermX method than there was with the DE method, which gives a better statistic for inclusion. I'll now pass over to Dr. Erin Hansen so she can talk about the University of Central Florida's STR results.
Thanks, Joni. Just for a reminder to everybody for the UCF portion of this study, for the conventional DE method, we had used the off-the-shelf, just standard recommended conditions for differential extraction using the EZ1. And we used that method along with SpermX in order to evaluate these 8 mixtures, and each one was done in triplicate. And the STR profiles were provided from one of the replicates. We just selected the best one out of the 3. So the first section of data that we're looking at here is an evaluation of the amount of unshared male alleles that were recovered in each one of these mixture samples. Because we had used global filer, like Joni mentioned before, there was a possibility of 32 unshared male alleles in our profile. So in this table are percentages of observed unshared male alleles out of the total possible. So for the SpermX method, you can see for 7 out of the 8 mixture samples, we had either full profiles or mostly full profiles with only one allele dropout. You'll see for mixtures C and F, there was one allele that dropped out, which would have given us the 100%. So for 7 out of those 8 mixtures, we got essentially full recovery of those unshared male alleles. And what was nice to see was 3 out of those were in the lower end sperm range mixtures, in those E to H mixtures. So that was really promising to see almost full male profiles for a majority of those samples. For the differential extraction, you can see in the DE column, the results were a bit more variable. We went from no recovery of unshared male alleles in that H mixture up to 94% recovery in the A mixture. So in between the range of samples, the amount recovered was pretty variable. You can see some pretty low percentages for some of these mixtures, and then kind of higher ones here in the middle. So overall, when you evaluate these percentages for SpermX, the average was 92% male allele recovery, compared to the differential extraction conventional method, which was only about 33%. So looking at about a 20-fold increase when you use the SpermX method compared to the conventional differential extraction method. This slide is a nice graphical representation of the data that we were just going over on the previous slide, and it's a little bit easier to see the trends in the amount of unshared male alleles that were recovered across this mixture set. Again, the blue bars representing the SpermX data, and then the striped green bars representing the conventional differential extraction method. And so what's really obvious from this slide is the almost, full profile recovery from 7 out of 8 mixture samples using the SpermX. And then again, you can see that large variability in the recovery of male alleles that we got using the conventional differential extraction method. One data point that kind of stands out in our data set is for sample B here, where we got very poor male recovery. We likely lost the sperm pellet in that sample, and I'm going to have to attribute that to my having to dust off my differential extraction abilities since I haven't used them in quite some time. So we'll just blame that one on operator error. The next two slides are going to show actual electrophoregram data from some of these mixtures that we've been talking about from our UCF set of data. So what we're looking at on this slide is the G mixture, which, just to remind everyone, is about a 4,500 to 1 female to male ratio. For the SpermX method, overall, we were able to observe 14 of those unshared male alleles out of those 32 possible. And then for the standard conventional differential extraction, we only observed 1 of those. And you can see that here in this top panel, where the only indication that a male was present in this mixture was here at DYS 391, where we were able to detect the presence of that allele. Just for comparison, we chose that same green channel, and these boxes that are highlighted in purple are the unshared male-specific alleles. It's a little bit difficult to see them on this electrophoregram because the scale is set so high, but it's just a nice comparison between the two methods. Again, we're getting male detection not only from DYS 391 here, but we have the indel, and then the Y is detected here as well when we used the SpermX method. What we're looking at on this slide are the results from the H mixture, which again is that approximately 18,000 to 1 female to male ratio. We tried to include a full profile here just because we always like to show more than just one channel. And this one just stood out because there's a very clear difference between the conventional differential extraction profile on the left and then the one that was recovered from the SpermX extraction. And you can see clearly in the SpermX extraction, you really get that minor male contributor. That's very easy to see. And you can see all the unshared male specific alleles are boxed there in those purple boxes. So this is one of my favorite pieces of data from this mixture study because it was the most significant mixture, and to get that many male alleles not only recovered but very easily discernible in this profile was really significant. So the next metric that was looked at for the UCF data was using StarMix to evaluate the male profile proportion in each one of these samples. Again, the bar graph showing SpermX in those blue bars and the standard differential in those green striped bars. And so overall, what we obtained from this data is that when we used the SpermX extraction, there was 2 to 36-fold higher male proportions in the STR sperm fraction profiles throughout this study. So again, just seeing that increase when SpermX was used compared to the conventional differential extraction. And then finally, what we're looking at here are those StarMix likelihood ratios that were determined for each of the samples. And again, just like we've seen on the previous slides, using SpermX resulted in higher likelihood ratios than the conventional DE methods for all of the mixture samples. And the fold differences that we were seeing ranged from 3 to 23 orders of magnitude, depending on the sample. And obviously having that significantly more male recovery in the SpermX samples really made it better statistics for inclusion overall. So just some overall final comments or perspective on this study from the UCF lab. This was actually pretty interesting when Joni first asked us to be a part of this study, because she asked us to use SpermX and compare it to our conventional differential extraction method. So for those of you that don't know, we're a research lab that tends to focus on challenging late reported sexual assault samples. And so pretty often we don't use a conventional differential extraction method. We work to develop advanced tools and techniques to use in those situations where conventional methods just don't work. Ultimately, our conventional DE method would be an organic extraction, which isn't often relevant to most labs since they've moved away from organic extractions for various reasons. So this study was a nice way for us to get back into some conventional DE testing and then evaluate a kit that could potentially be more helpful in some of these challenging sexual assault samples that we work with. And overall, the results from our portion of the study is really promising. The improvement in male DNA recovery and profile detection with the SpermX kit is significant, and especially in those low-end sperm mixtures, those E to H samples. And so hopefully, this can improve the analysis of sexual assault evidence and provide that much needed upgrade to conventional methods, like Joni had mentioned before. We're using methods from 1985, so perhaps this could be the nice upgrade or refresh for differential extractions that the community needs.
Thank you, Erin. We'll now hear from Tina Nichols from CFSRE on the STR results they obtained.
Thanks, Joni. As previously mentioned, our conventional differential extraction method included a few modifications from UCF's protocol. These changes included the use of our in-house EZ1 extraction buffer instead of the epithelial digest buffer. We also reduced the epithelial incubation period to 40 minutes and increased the sperm digestion period to 40 minutes. Our samples were also run in triplicate for the SpermX and conventional differential extraction method. These samples were plated in duplicate onto a quant plate, which provided us data including the small autosomal to Y ratio for each sample. Using this data, we were able to determine which of the triplicate samples from each method and set would continue on to amplification via globofiler and STR analysis. This led us to generate 16 STR profiles, 8 from each method, either SpermX or the conventional differential extraction, and 1 from each sample set A through H. The chart on the right is displaying the CFSRE's recovery of unshared male allele data. For both methods, you can see full profiles up until swab set E. While SpermX is still able to achieve a full unshared male allele profile recovery up until set G, sets E and F are where we begin to see the performance of the conventional differential extraction method decrease to 75% and 81.3% recovery respectively. The SpermX method was still able to achieve a 93.8% recovery for set G, while the DE method fell to 15.6% recovery, which for 32 alleles is only 5 alleles being detected, resulting in a 6-fold increase for SpermX. Set H, our most discrepant female-to-male mixture ratio, resulted in a 56.3% recovery for the SpermX method, while the DE method resulted in a 15.6% recovery, leading to a 3.6-fold increase for SpermX. On average, the SpermX method resulted in a 3-fold increase in percent recovery for unshared male alleles with a range of 1.2 to 6-fold higher recovery percentage. Overall, though both methods performed well in sets A through D, the low sperm series sets E through H really demonstrated how well SpermX can capture the sperm cells even with few present. Here we have the data from the previous slide displayed in a graphical format. The SpermX method, shown with the blue bars, achieved 100% recovery for 6 out of 8 sets, while the conventional differential extraction method, shown with the green striped bars, achieved 100% recovery for 4 out of the 8 sets. With this, you can really see the sudden drop in performance of the DE method for sets E through H, whereas the SpermX method was able to remain relatively consistent, with 93.8% recovery for set G and still achieving over 50% recovery for set H. Similarly to Joni and Aaron, I will be showing some of the STR data that the CFSRE generated during our study. I'm also going to be focusing on sets G and H, as I feel that the results from these sets really speak to SpermX's ability to generate usable results even at such discrepant female-to-male ratios. Just as a reminder, set G had a female-to-male mixture ratio of 4,545 to 1, with 1,500 nanograms of female DNA and 0.33 nanograms of male DNA. All of these samples were amplified using Globofiler, and displayed here I have the blue dye channel from both methods for set G. The circled alleles represent unshared male alleles that were detected. For this dye channel, you can see there were 5 unshared alleles, all of which were detected by the SpermX method. None of these 5 unshared male alleles for this dye channel were detected using the conventional differential extraction method. When examining these profiles as a whole, the SpermX method resulted in the recovery of 30 out of 32 unshared male alleles, while the conventional differential extraction method resulted in the recovery of 5 out of 32 unshared male alleles. Moving on to set H, we have the purple dye channel from the Globofiler kit. As a quick reminder, set H was the most discrepant female-to-male ratio tested for the study, with a ratio of 18,182 to 1, containing 1,500 nanograms of female DNA and 0.0825 nanograms or 82 picograms of male DNA. Similarly to the previous slide, I circled some of the 32 unshared male alleles that were present in the SpermX method samples on the left. Though all 5 of the unshared male alleles for this channel were detected using the SpermX method, the conventional differential extraction method, shown on the right, was not able to recover any of these 5 alleles. Though I am only showing the purple dye channel here, when analyzing the entire profile from each method, the SpermX method was able to recover 18 out of 32 alleles for set H, while the conventional differential extraction method resulted in the recovery of 5 out of 32 alleles. Some other notable points about these profiles for set H. When examining the amylogenin Y-indel in DYS391, the Y allele was not detected at amylogenin, the 2 was not detected at the Y-indel, but the expected 11 allele was detected at DYS391 for the conventional differential extraction method. The SpermX method resulted in the recovery of the Y allele at amylogenin, 2 at Y-indel, and the 11 allele at DYS391. As previously mentioned, we were able to analyze the male profile proportion for our samples using StarMix software. Like Joni and Aaron's graph, the SpermX data was shown with the blue bar, while the conventional differential extraction data was shown using the green striped bar. The chart on the left displays the numerical values for the graph on the right, which can be helpful for the lower values. As you can see from this graph, the SpermX method achieved a greater male profile proportion than the conventional differential extraction method for each set analyzed. For example, SpermX was able to reach 86.1% male DNA in the STR profile, meaning roughly 14% of the profile was attributed to female DNA, compared to the conventional differential extraction method, where 38.6% of the profile was attributed to the male and the remaining 62% attributed to the female. On average, between the two methods, SpermX was able to achieve a 5.7-fold increase, with a range of 2.2 to roughly 24-fold more male in the sperm fraction STR profiles. The last aspect of the study that we will be looking at for the CFSRE's data are the likelihood ratios, which were able to be calculated using StarMix software. For our data, the analysis of samples A and H resulted in comparable likelihood ratios with a fold difference of approximately 1, meaning that the SX and DE methods' likelihood ratios for both of these sets were essentially of equal strength. Looking at the remaining sample sets, the fold difference between the method's LRs ranged from 2 to 17 orders of magnitude. Overall, this was able to demonstrate that because there was significantly more male present in the SpermX profiles, these samples resulted in much more favorable statistics for inclusion. To finish out my section, I thought I would summarize with some of our final thoughts after completing the CFSRE's portion of this study. Overall, from our data, it was clear to us that the SpermX method outperformed our conventional differential extraction method through the improved male DNA recovery and achieving more complete male STR profiles. Looking at the SpermX device as a whole, it is relatively simple to use and takes a lot of the variation between analysts out of the equation. This leads to more consistent results and less hands-on time during analysis. Looking towards the future, we feel that this could be a new and improved method for testing sexual assault kits, especially with samples containing low levels of sperm cells. Overall, it's very encouraging to see. And with that, I will hand it back to Joni.
Thank you, Tina. I'll now summarize the quantification metrics. The male DNA recovered, SX recovered 5 times more. The SY ratio and the sperm fraction, the DE ratios range from 5.4 to 178 times higher than SpermX. Percentage male and the sperm fraction relative to theoretical. SpermX was 1.5 to 10 times more for the ample DNA series, and 20 to 33 times more for the low sperm series. Female carryover into the sperm fraction was higher with the DE method, and how much higher? About 2.8 to 10-fold more carryover than the SpermX method. In conclusion, all quantification metrics to evaluate the effectiveness of a differential extraction method showed that the SpermX method outperformed the DE method. The STR analysis showed higher recovery of unshared male alleles. All 3 labs gave similar trends for all the mixtures, exhibiting good reproducibility across the laboratories. And this study showed that the SpermX method captures more male DNA with less female carryover, which results in a more informative STR profile than the conventional differential extraction method, such that sexual assault kit evidence containing semen should be processed using the SpermX method to obtain the most informative, probative male profile from evidentiary type samples. And this is especially true for swabs containing low sperm amounts, which is often encountered in forensic casework. The use of the SpermX method can have a great impact to the forensic community. As we know in forensic cases, victims do not always go to the authorities right away, and sometimes they bathe or are forced to douche, etc., which can lead to low sperm. We've seen over the years that the percentage of sexual assault cases that don't have enough DNA for comparison to a known or for CODIS upload can range from 30 to 50% or more. The SpermX method can help solve cases that would have been reported as insufficient DNA for analysis heretofore using the conventional differential extraction method. And I'll bring your attention to the quality assurance standard 7.3.3, which actually resides in the evidence control section of the standards. But we have shown in our validation that the current DE method has a loss of up to 80% of the sperm when you remove the swab. So if I read the standard, it says the laboratory shall have and follow procedures that address handling and preserving the integrity of evidence and work product designed to minimize loss, contamination, and/or deleterious effects. I would say that we should look at this and the fact that we have not gotten a lot in our conventional differential extractions in terms of the amount of sperm that is actually on the swab, that we really need to pay attention and change our methods that have been around since 1985 or so, such that we can get much better results from our evidentiary materials as forensic scientists. And in that sense, SpermX can be used to capture sperm from very low evidentiary samples, such as swabbing of microscope slides, oral swabs, and other items that may not be successful, or some labs may not even attempt with the conventional differential extraction methods. Thank you all for attending today. I'd like to draw your attention to the fact that the research was funded by an NIJ grant for SpermX research and development, and I've listed the contact information for Dr. Sudhir Sinha and myself, and we can now open it up for questions.
Alrighty, thank you all for providing such an informative presentation. Our panelists are now going to take audience questions.
All right, everyone, we have a bunch of questions in our box here. There's several for me, so I'll answer those first. One of the questions is, what's the threshold used to get the allele count? I'm assuming that's the manual unshared male alleles. And that was actually shown in the presentation where the thresholds for UCF and our cells were 100 RFU, and for CFSRE, it had been 40 RFU. And before I answer all the questions, we have Dr. Jack Ballantyne joining us, and I'll let him make a few comments on these studies. Take it away, Jack.
Thanks, Joni. I hope everyone has enjoyed the webinar. I certainly did, and learned a few things since I hadn't seen all of the data from all the labs. But standard differential extraction, of course, has stood us all in good stead over the years since it was introduced in 1985. And any self-respecting lab doing sexual assault investigations will be employing differential extraction. But we all know, those of us who have worked in this business for a long time know there are limitations to standard differential extraction that the studies that have been presented have illustrated. And basically, the limitations are incomplete separation of the sperm and non-sperm fractions, as occurs quite often, and significantly there's a loss of sperm. Many studies have shown, including this one, that you lose sperm if you use a standard differential extraction process. Looking at the data from the developmental validation that InnoGenomics has published, that Joni mentioned at the beginning of our talk, plus the studies that we participated in, basically SpermX does show good potential, I would say, to significantly improve the efficiency of that DE process. So my comment would be, if I was running a lab right now, I would consider investing in validating this system because the benefits seem to be so great. But it remains to be seen, of course, in that other labs have to reproduce this using your own analysts, etc., with your own work, your work procedures, workflows, etc. But I would consider validating this system to see whether or not these benefits can be applied to your own casework. So that's my comments. I think it's a very promising system based on what I've seen so far. And lastly, I should say, it hasn't been mentioned, I don't think, that looking at the way the system was developed, the developers, InnoGenomics, obviously there's a nod to automation down the road because the consumables, the tubes that are used to perform the manual studies that we did here are the same tubes that would be used basically for robotics. So I think I haven't seen any of the data yet for the automation, but this system is apparently readily automatable. So that's another thing to consider as well. So that's my thoughts, Joni.
Okay, thanks, Jack. All right, Evan, Aaron, there was a question for you that maybe I could answer. It was, were the swabs re-extracted after the initial extraction to see if there's any DNA left on the swabs? This was not something that we put out to the other collaborators for this study. We did do that in our validation that's published. And generally, we see about 80% of the sperm DNA from the conventional differential extraction remains on the swab that gets removed after the first epithelial digest. So that's very concerning to me. Some old studies, we didn't do that with the SpermX swabs, but some old studies show that it's about 0.5 to 15% is what we saw remaining on swabs that get re-extracted after the process, to answer that question for you, Aaron. Another question for you, Aaron, is what were your amplification targets, the nanograms in total?
We tried to target 1 nanogram, and for most samples, we were able to achieve that. There were 2 on the standard conventional differential that we had 65 to 200 picograms, and that was the max we could put in. For the SpermX, we had 4 samples that were under the 1 nanogram target, but they were all 700 to 900 picograms, so they were pretty close to our target input.
Okay. Another question for you, Aaron. Is there any idea as to why group G had lower recovery percent with SpermX compared to group H, which had a higher female to male ratio?
Yeah, I don't have an answer for that. G just kind of was a little bit of an outlier compared to all the other ones. So I think if we could go back and repeat that one, we might have a different outcome. It just seemed to be a one-off kind of issue with that sample.
Okay. And for you, Tina, how long was the SpermX process versus the differential extraction process?
Yeah, so the classical differential process probably took me about 2 to 3 hours per batch, just because we had that longer incubation of 40 minutes. The SpermX process in total probably took like 7 hours, but it's a lot less hands-on than the traditional process. So I could do other things during that because most of that time was just incubation.
Okay. There was a question for me. Were there any drop-in or background peaks that needed to be curated out? So as far as edits in the STR profiles, we had no drop-in whatsoever. We did have artifactual peaks like stutter and things like that that needed to be edited out, but there was no contamination or drop-in peaks to answer that question. Okay, now this is for Erin and Tina. Did all conventional differential extraction methods only incorporate a single epi lysis step? If so, do you think the additional epi for the SpermX contributed to the improvements observed?
I guess I can answer that first, and then we'll see if Tina agrees with me or not. Yeah, I mean, I definitely think, that second lysis step is helping get rid of some additional epi that might be stuck on that swab. So it would be an interesting idea to try that in a conventional, differential extraction to see if that had any difference. But I mean, to me, I think the overall impact from the SpermX is the fact that the swab is in that sample for the entire extraction process. So before, with conventional methods, we take it out after that first epilysis. And so I think some of the improvements we're seeing is that the swab is in there for that whole time, and kind of you can get more material off that swab, hopefully, by having it in there that whole time.
Yeah, I definitely agree with what Erin said. We also only had one epithelial and then one sperm digestion. So I would be curious of what would happen if you added an additional one as well.
And I'll just chime in on that, because what we tried to do here is try and simulate what most laboratories are doing. So there, as far as my knowledge, not many labs are incorporating a second epithelial digest. So we were using a lot of what manufacturers put out in kits as their separation method, and what most labs do today. So if there are more labs doing secondary epithelial lysis, I'll apologize, but we tried to simulate what's being used now versus the new method. And as they said, of course, I think a second epi digest, I know California Department of Justice incorporates that, and maybe a few other labs. So that will help somewhat, but the real key here is the nanofiber membrane that's going to trap those cells, and then you don't have any issues with the separation and the technique doing that, and the fact that that swab stays in. As we've shown in the validation, 80% of the DNA is lost in the removal of the swab. Back when I was at Massachusetts State Police Crime Lab and other labs, we sometimes would go back to the substrate and extract that too if we had a really important sample that didn't have enough. But that was very rare that occurred. And now you can leave it in with SpermX all the time. Okay, on time, we have still another 5 or 6 minutes. So one of the questions that was here for me is, how many different donors of sperm and E cells were tried? I guess in this study, it was one set of one female and one male, but at InnoGenomics, we've tried many dozens of different contributors to answer that one. Let's see. Are there any plans to compare it to an automated differential process like the QIAcube? Well, I could say that we have Utah Bureau of Forensic Sciences has done work, and their current method was with a QIAcube. They've presented at ISHI in the past, and we can get that data per request that there was comparison of Utah's differential process was using our SpermX device, and at that time they were using their own buffers, and they did comparisons of data between the two, and their current method was with QIAcube. So I can get more information or put you in contact with the folks at Utah as needed. Thank you for the question. All right. Here's another one. Was STR mix analysis run according to known data about the number of contributors, or was the data analyzed by analysts who utilized the threshold to assess the data? This was put in as a 2-person mixture into StarMix. It was identified as a 2-person mixture only. As I mentioned, no drop-ins. Dr. Tim Califett did all the work on SpermX, and he put that in. And he had the known male, as we said in the presentation, he had the known male and female as the knowns, and he used these samples as the evidentiary samples. Let's see, what other questions we have. One of the questions, have you determined a quantity or concentration of DNA upwards of which you can see carryover of female DNA in the sperm fraction? Well, depending on the mixture and the ratio, even with these samples, we did see female carryover into the sperm fraction, just that it was carryover such that you could still pick out either a fairly major male for the A sample or other ones that were from the B series down, whereby you had a minor male contributor. I think Erin could probably answer to that also. She spoke about her STR results to me personally, so I'll let her share them with you.
I mean, we don't have like a specific, quantity or concentration where you would see that carryover. So we've never actually empirically determined that. I don't know if anyone in Tina's lab has done that.
Not to my knowledge.
Okay. All right. A few more questions. We have 3 minutes here. One of the questions is what equipment and reagents are needed for the lab to use SpermX? And in that regard, it's simply most the kind of equipment that's in the laboratory. You would need a centrifuge with certain accessories. We generally use an Eppendorf. It's a 5810R model. There's a swing bucket with MTP buckets that we use. And there's a special Autolus M rack. The rack that actually holds the samples is a small gray rack that we actually provide with our kits. I don't know if you can see this that well, but here is the rack. It's a gray rack, and that has some of the tubes in it, that the blue are the manual and the orange are the automated. So basically it's a setup to be able to put this into an adapter that then goes right onto the centrifuge, and that's how you spin those samples. The other reagents that you would have to provide would be things like Pro-K, DTT, and to have some sort of thermal mixer. We actually have done a lot of work with the Eppendorf Thermal Mixer R or the Eppendorf Comfort. There's also a Mixer HC that's also used to give you some of the specifics. And then another one says, are there trial kits for the labs to make an evaluation of SpermX? And the answer to that is yes. We provide a trial kit that normally allows for the processing of 20 samples. And we try and supply swabs, sample swabs with that, so you can practice with them. We give test swabs that are very easy ratios, just so people can practice, make sure it's working in their hands. And we are starting to prepare and provide comparison swabs for method comparison. And what we're doing is we're replicating, and it may be with different donors depending on our sources, but we're in the midst of replicating the E through H series so that you can do it in your own lab. After you've done practicing, as Tina and Dr. Hansen did, you can go ahead and run these in your own laboratory. So I think, unfortunately, we do not have time for all the questions, but please feel free to reach out to us for additional information. The contact information will reappear in the final slides. And thank you all for attending today.
Thank you.
Before you leave today's webinar, please participate in our short 5-minute survey. Simply click the checkbox icon directly above the chat to access the audience feedback survey for today's event. Your feedback is critical information that helps us improve our webinars and continue providing the best experience possible. We would greatly appreciate it. Lastly, across the top of the live stream window are links to our social media profiles, including Twitter, LinkedIn, and Facebook. There is also a link to the FDCOE website, the Just Science podcast, and the FDCOE newsletter registration. Like, follow, or subscribe to stay up to date with everything the FDCOE is doing in support of the forensic community. Thank you again for attending, and we hope everyone has a great day.